Thank you very much, and it's been a fantastic meeting. The scientific meeting the last couple of days has been really amazing. Now I'm going to talk about B-cell biology, and seeing it's after lunch, I promise to be gentle, but I think it's important because you've all heard of Rituximab, and I think it's important to have a platform to be more informed about what B-cell depletion means, and to know what it means, you need to know about B-cells. So I work in the rheumatology department at UCL with a consultant rheumatologist, and I worked for many years, more than I care to think about, with Professor Joe Edwards. And as you probably all know, Professor Edwards was the first one to use Rituximab in autoimmune inflammatory diseases, and that was way back in 1998. And it was based on a hypothesis we developed at UCL. It was all published and before we treated the patients, but it still took at least 2 years to get the paper published on the first patient. So I'm familiar with problems. Okay, so the main work that I do is in B-cell biology and autoimmunity, particularly in rheumatoid, lupus, myositis, and I've done a bit of work on pregnancy as well. And we focus our work on biomarkers for a clinical response to Rituximab. So we're interested in identifying the patients who will respond best to Rituximab, and also predicting relapse, because patients don't like relapsing. They don't like getting their disease back in a worse form. We're also looking at other B-cell directed therapies against the same molecule as Rituximab's against, and other ways of changing B-cell function. So why Me Cfs at UCL? Well, we were very excited by the placebo-controlled trial published in 2011 by Drs. Flug and Meller from Norway, and we've been collaborating with them ever since, actually. So we've obviously got an established interest in Rituximab and B-cells at UCL, so it's working very well. Okay, so this is a B-cell, and as you can see, it's got lots of different markers on it. This is about 25% of all the markers on B-cells, so they're quite complicated. When I first started at university, I remember lymphocytes were small round cells of unknown function, so things have changed a little since then.
I didn't know you were that age.
I am. We had very old books in Australia. Okay, so a lot of these markers, I'm not going to go through them, don't worry. It's going to be very gentle. A lot of these markers can tell you how long the B-cell has been out in the circulation, whether it's experienced antigen or not, whether it's come in contact with a T-cell or an NK cell, a mast cell, a neutrophil, all kinds of things. And then other markers on the B-cell are involved in adherence. And basically, the immune system is just one big mess of Velcro of things sticking together. So they're very important molecules that control whether the cell's going to stick to the spaces between the cells or the matrix proteins or other cells. But the most important thing about the B-cell, of course, is the B-cell receptor. And the B-cell receptor is also known as antibody, but it's bound to the surface of the cell. And so every new B-cell exiting the bone marrow has a different B-cell receptor, and you'll be pleased that I'm not going to go into the genetics of that. So every B-cell is different, and every day about 10 to the 9 B-cells come out into the circulation, so that's quite a lot of cells with different receptors. Now, when often you sort of say, oh, B-cells make antibodies, it sort of seems to be the obvious thing, but they don't. They make very small amounts of antibodies. The thing that make antibodies are plasma cells, which the B-cells turn into. And these are just big bags of antibodies, but they don't express it on the B-cell receptor on their surface anymore. So it's the things that change the B-cell into a plasma cell making antibodies is obviously the basis of B-cell biology. So antibodies are just the soluble form of the B-cell receptor. So the B-cell receptor on the B-cell, when it's made in large quantities, and I know this slide is wrong, but I don't want to draw another plasma cell. And these antibodies, as you know, circulate in the blood and they're released onto surfaces. In fact, most of the antibody made in the body is actually secreted onto the surfaces, the gut in particular, the lungs, mucosa. So there's a lot of it that you don't see in the serum. And as I'm sure you know, their main function is to bind to and help to clear microorganisms from the body. A lot of the antibodies that we talk about, we'll just go through where they come from. B cells are made in the bone marrow. Oh, back. So they exit the bone marrow, these new B cells every day, but they only live for a little while. If they're not stimulated or kept alive by various factors, then they circulate through the blood into the lymphoid organs. And I'm sure you've all known that when you get an infection, your lymph nodes swell up. And that's because the lymphocytes are trapped in there. The whole thing about the immune system is it's contextual. Things don't happen floating around in the blood. They happen in a certain space under very strict rules, if you like. And when those rules go wrong, you get things like autoimmunity, for example. Okay, so each B cell receptor or antibody recognizes a different shape on a target bug, or as I keep calling it, antigen. So antigen just is a bit of bug. It's usually in a three-dimensional shape, so it recognizes shapes. So an antibody to tetanus, for example, will not recognize flu virus. And antibodies come in different shapes and sizes. Now, they all share the same thing of being able to bind antigens, which are coming in here in blue. And there are actually 5 main different kinds of antibody, but I've just shown 3 here. The IgM one, as you can see, it's got 10 binding sites. IgA's got 4 and IgG 2 for the same antigen. Each arm recognizes the same antigen. And they're very different because of not the antigen binding end here, but this bit here. This is called the Fc bit. This is IgA, which is particularly made for being secreted onto surfaces. This is actually made in the most abundance in the body, even though in serum, IgG is the most abundant. IgM, I'll talk about a little more in a moment. And these structures at the other end of the binding site have unique properties. There are, at least four kinds of IgG as well. And this end here, and the different 4 kinds of IgG alone, will bind to cells in different ways, bind to other components in the blood in different ways, change the function of the cells they bind to in totally different ways. IgA is particularly suited to being spurted out, if you like, onto mucosal surfaces because it's got a secretory piece. IgM is very good at binding bugs, and when it changes shape, it's extra good at being cleared from the body. Okay, so the main thing about antibodies, of course, is that they bind antigens, and this is unbelievably complicated, this whole maturation process. So I'll simplify it like this. B cells come out of the bone marrow, start circulating around. When they encounter antigen, the first thing they'll do is they'll make a little bit of IgM. And then if they come across the right sorts of cells in the spleen or lymph nodes or other places sometimes, they will transform into plasma cells and make IgG. And IgG really is the most high affinity. They'll also undergo what's called affinity maturation. They will be the most high affinity antibodies will be selected by binding to the antigen. So these ones here are a better model of fit for a particular antigen than these ones here, although these are extremely important. Okay, so when you encounter a bug, what happens is, floating around in your serum anyway, there are sort of low affinity antibodies, and these can be a bit of a problem and can cause things like infusion reactions sometimes because they're sort of pre-formed, and if they happen to recognize some drug or something that by chance, they can cause some problems, but that's rare. Mostly when you come across a virus or something here, you get IgM produced, and then by this affinity maturation, you get IgG. And this is a classical secondary response. The other important thing, which I hope is on this slide, oh no, it isn't. You know, the other thing about these cells that make this here is that some of them turn into plasma cells and make antibody. The other ones turn into memory B cells. And these memory B cells are in larger number. So when that bug comes back again, there's an army already pre-formed, if you like, to attack the bug. And as I've said, the other end of the antibody molecule can bind to cells, for example, a macrophage. And the macrophage, when it's stimulated by the antibody bound to the bug, can produce toxins and help get rid of the bug. Okay, so just to summarize the life cycle of the B cell, you've got B cells continuously leaving the bone marrow and entering the circulation. And then they go through these secondary lymphoid organs in the skin, etc., and look around for antigens. And then they recognize, and then they interact with other cells, form plasma cells to make antibody, and high-affinity antibodies and memory cells. Okay, so as I've said, the B cells turn into plasma cells to make the autoantibody. But in order to get all those other things I've talked about, they need to interact with other cells. And the most usual way, the most important way, is through their interactions with T cells, which I'm sure you've all heard of. So the B cell recognizes an antigen and it shows it to the T cell. I'm certainly not going to go into this, it's too complicated now. The T cell has already experienced bits of this antigen because of the way the cells get together in this particular environment. The T cell makes cytokines, which helps the B cell make antibodies. So this interaction is crucial. Now, interestingly, we've heard about cytokines today. Now, in the case of Me Cfs, there are lots of reports of cytokines being up, down in different papers. There's been a lot of controversy about it. But what we've taken note of are two really nice studies, one by Maddie Hornig and one by Broderick et al., where they've compared, as we were talking before, groups of patients who got Me Cfs after having an EBV infection and another control group where the patient had an EBV infection but recovered. So, and there they found that these cytokines were increased and this one was decreased. So those sort of studies are so useful for us because then we get a really clear idea of what we should be looking at. And Dr. Hornig showed that if you look at early versus late disease, again, you've got different signatures, really useful data. That's the sort of stuff we need to work on. There are other kinds of B cell antigens or antigens recognized by B cells which don't need T cells, and these may be equally important in some infections, particularly to bacteria. And in this case, the antigen is sort of big with lots of different repeating bits in it, so it can pull together a lot of these receptors on the surface without much of a need for T cells, because it produces enough signal, and you can get antibodies, but they produce memory cells, but not those high-affinity IgG memory cells. They're just IgM ones. Okay, so what about B cells and autoantibodies or antibodies in disease, and how can this possibly relate to Me Cfs? Well, the answer is I don't know, because the evidence for autoimmunity, as I understand it, is fairly small as yet in Me Cfs, but from experience in other diseases, antibodies to self can take a lot of different forms, and they can just, in the form of complexes, activate cells and cause inflammation. It doesn't really matter what they're against. As long as you make enough antibodies and pick up bits of self, you're going to drive inflammation. And that happens in lupus and rheumatoid. Antibodies can also bind to, autoantibodies bind to and change the function of cells. In a kidney disease called vasculitis, there are antibodies actually against neutrophils, the inflammatory white blood cells. And, of course, other antibodies interfere with the communications between cells and receptors, like against acetylcholine receptor, muscarinic receptors in Sjögren's, for example. There are many, many examples, but I don't want to go too much into other examples. We need to think about Me Cfs. Right. Okay, now just a few slides about rituximab. Rituximab works best in diseases where there are autoantibodies. There's a very long list now of diseases where rituximab has been tried. If you look at those, they've all got, or most of them have got an antibody, an autoantibody associated. So antibodies which are known or even suspected to be involved in the disease are best treated with rituximab. So rituximab recognizes an antigen on B cells called CD20, and when it kills the B cells, of course you'll decrease antibody production, cytokine production, virus-infected B cells. EBV is cleared by rituximab in one report. And it'll get rid of overactive or dysfunctional cells. And really, unless you've got a feedback mechanism keeping them going, it can produce remission for some time. So rituximab removes all of the cells out in the circulation. It doesn't get rid of those plasma cells that make antibodies, and it doesn't get rid of stem cells. So you can regenerate your B cells. So there's no real obvious harm. Okay, so I'm just going to show you a couple of slides about how you look at what antibodies do after rituximab. All right for time, Ian? Yes? Okay. Great. Okay, so I'm just going to show you, as I say, a couple of slides because it's quite tricky. You think, oh, well, antibodies cause disease, they'll go down after rituximab and then they'll come up when the patient relapses. Some do, but then not always. Now, this is just a heat map looking at antibodies to 52 different bits of an autoantigen called a citrinated protein. And these are the responses of 16 patients through a course of rituximab. So I won't give you any more of that. I'll just show you what happens. So using those that multiplex array, what you can do is measure the significance of the binding, here using this Z-score, which again I won't go into. And then what we do at UCL is look at 3 different key points in the cycle of rituximab treatment. Pre-treatment, when the patient's depleted, and when they relapse, because patients differ very much. So we've got, functional points, not time points. And you can see that the levels of these antibodies, you have to know what they are, they go down, but then at relapse, they don't seem to come back. But when you look at these antibodies in detail and follow individual ones, you can see that some go up, some go down, and if you separate those ones out, you can see that some of these antibodies go down and up beautifully, significantly at relapse. Some of them go down and disappear, and some actually go up and down, about 25%. So it's complicated, very complicated. But it gives you some idea of how different clones respond to different depletion techniques. Okay, now finally we're going to get on to Me Cfs. And for those who are at the meeting in the last couple of days, our PhD student Fane Mensah presented this data. He's worked very hard on this for the last year. And some of you in the audience will know Fane very well. He's taken your blood with the assistance of Arti Sharma, who gives her time free to help with the project. So I'm going to give a very truncated and hopefully accurate assessment of Fane's data. Just talking about B cells and Me Cfs, there's really no clear picture. We've looked at all the literature, and the papers differ for the obvious reasons. I don't need to tell you, you know better than I, how difficult these things are to get a homogeneous population. So the approach we took nearly a year ago now was to compare cohorts from 2 different centres, one of them, of course, Dr. Bansal, who you saw this morning, and a consultant physician at UCL, Dr. Saul Berkowitz. And the question we asked ourselves initially was, is it possible to stratify these patients on the basis of their B cells? Because it's just the change is going to be so smallish, we think, if you look at Professor Hornig's data, and she agrees, that looking at healthy controls versus patients is not really, it's just too controversial, really. So we also aimed, therefore, to look at B-cell phenotype and demographics. The study design is very simply is that Fane sends out the patient information packs, the patients visit us, fill in the consent forms questionnaire and the blood sample taken, and we measure B-cell phenotype using flow cytometry. Now, again, I'm not going to go into this, but each of these markers, you can buy monoclonal antibodies to each of these, and they all come in all different colours. So you can do combinations of, I think Fane uses 8 different ones per tube. So you get a very complicated picture. And again, they're against those different kinds of markers. Right, so this is a typical FACS plot. I won't go into it. Each of these dots here is one cell. And what you do is you look at how bright each cell is with this marker or this marker. And these ones here, these CD27 high ones over here, fall into 2 types, and these young ones here fall into 2 types here. So you can do quite complicated things, and Fane's done a nice diagram here showing you can look at the maturation. We'll just do the top one here. If you look at naive cells, they've got this orange one, but no blue one. The medium ones have got both. The switched memory cells have got just the blue one, and these ones here don't have anything. So you can get quite a lot of information out of FACS analysis. The other analysis we use looks at additional subsets, the transitional cells, the very early ones come out of the bone marrow, and also plasmablasts, which are just about to become plasma cells. I don't know if you can see this, but we looked at 38 Me Cfs patients and 32 controls. We just looked at the percentage of B cells in the blood, comparing healthy controls with patients, and this is normal. So the patients had the same percentage of B cells as normal controls. The other things that we looked at were the percentages of each of the memory and naive phenotypes using both those classifications. We found no differences. But then Fane looked further at another marker, again, I'm not going to go into it, called CD24. And CD24 is present on young cells, and so he's looked within all the populations of B cells. This is looking at the CD24 on total B cells, and you can see that significantly higher percentage of cells with this marker, and also it's significantly brighter. It doesn't look much here, the differences don't, but that's quite significant for this marker. This particular antigen is present on early B cells at the highest amount, so it's present on young B cells, and it's gradually lost over time. This is maturation, early stage, late stage, so it gets lost. It's quite an interesting molecule, but it's mostly associated with young B cells that haven't yet encountered antigen, and it may regulate how the B cell interacts with antigen. Okay, and the other population that we found differences in was this CD38, CD21 positive subpopulation, which is a memory subset. And using statistics, they were stratified into higher normal expression in the patients. And what we found was that low expression was highest in normal controls. So if you're a normal control, you'll have like low expression of a low percentage of these B cells. And the ones with the high will be fewer. But if you look at the patients, you can see there's about equal numbers of high and low CD21, CD38 positive B cells, negative B cells. And the other thing was stratifying, we're looking at demographics, when the patients were stratified into those who had less than 5-year duration and greater than 5-year, and this is number of patients here, you can see that the patients with it's the last slide, the lower disease history, there were more of them compared to the ones with a longer history, and these ones look more like normal controls. Right, so just to summarise, we found no differences in the classical B set subpopulations. A higher CD24 percentage in MFI, that means expression in the patients compared to the controls, and it was confined to the young B cells. We also found differences in memory subpopulations too, which were related to disease duration, and we've been looking at another population of innate B cells, but I won't present that data, obviously, because I've got time. So the future plans of Fane, and he's better at expressing them than me, is looking at functional assays. We're working very closely with our Norwegian colleagues, and we want to look at functional tests on stratified populations. So I'd like to thank my colleagues at UCL for being supportive, very supportive, and of course I can't thank Richard and Pierre enough for their support and putting on the most fantastic conference every year. Thank you very much.
Thank you. Okay, we have time for a question. Yes, Angela.
When you showed the post-Rituximab relapse and remission after, you showed some measurements. That was total IgG you were looking at, or what? Was it a specific antibody?
Oh, it was anti-CCP antibodies.
And what are the subclasses of those antibodies?
I only looked at, I've done IgG and IgA. IgA, you don't
subclasses, IgG
Oh,
subclasses.
No. I've only done, because it's multiplex, you can only do really G and A, and they can't do the G subclasses, and they can't do IgM.
Because in myasthenia gravis, which some of the audience might remember from my talk last year, is caused by antibodies. There are two antigens, acetylcholine receptor and another one called MuSK. And the IgG1 antibodies to acetylcholine receptor really don't do well after rituximab.
No.
Whereas the IgG4 antibodies to MuSK actually go down and the patients get considerably better.
Yes.
So it could really be interesting to try and see what yours are.
CCP. I've done it with just the conventional antigen, but not to the other 32 proteins. But I would love to.
Okay, Joe, many thanks.
Thank you.
Sorry, is that another question? Sorry, there's another one.
Joe, can it shortly... Hello.
Oh, sorry.
Joe, you mentioned in the beginning the mechanism involved in vasculitis and immune complex activation of complement system, as well as blocking the transfer signals between brain and signals in the nerve. I wonder, have you looked at complement in respect to the disease here? Or somehow,
I
will
haven't,
you comment upon the phenomenon that we do not have that much results on complement activation?
No.
Is it due to clonal differentiation?
I don't know. I mean, there's the data. I don't know if Joe has anything to add, but I mean, as far as I know, people have looked at the complement components. Like C4 would be interesting, but then the level doesn't have to be low. If you've got a null allele, it doesn't function properly, even though the levels are okay. I don't, I would like to, but not all complexes fix complement either. So, something
Joe.
we'd like to do. Thank you.
Many thanks indeed.